Now we will talk about angiosperms. Angiosperms are the flowering plant, and unlike gymnosperms, they are having seeds which are covered with ovary wall. Since they are flowering plants, that means they are going to have flowers, and these flowers have two kind of accessory organs, that is the male gametophyte and female gametophyte. The male gametophytes are having a filament and an anther. These anthers are bilobed, and when it gets matures, it will release pollen grains. While in the case of female gametophyte, they are going to have ovary, then style, then stigma. So this is the general structure of female and male gametophyte. So now how these fertilizations take place? This fertilization take place with the method of pollinations. Pollinations, you know what is it? It is the transfer of pollen grains from one plant to another plant. These pollen grains are accepted by the stigma and they will form a pollen tube. With this pollen tube, they will carry the male gametes and it gets fertilized to form zygote. Angiosperm have two classes, that is dicotyledons and monocotyledons. Dicotyledons are the one which are having two cotyledons and the monocotyledons are the one which are having single cotyledons. Well, cotyledons, that means a leaf-like structure which can be seen while germinating a seed. Inside this ovary, there contains ovules and these ovules will form embryo sac. This embryo sac is a 7-celled 8 nuclei. I will discuss about the 7-celled 8 nuclei in more details. Let me first tell you what this embryo sac contains. This embryo sac contains 3 cells, that is in the micropylar region, which we call as antipodal cells, and next is the central cell, which is the polar nuclei, and the next is synergid, these two synergids. With this, they form the structure of the female gametophyte. Now let's see the life cycle of this angiosperm. The plant body is known as sporophyte. It is going to produce two kinds of gametophyte, the male gametophyte and the female gametophyte. The male gametophyte is giving rise to the microsporangium and the female gametophyte is giving rise to megaspore mother cells. Microsporangium is dividing by meiosis and it produces microspores. These microspores are haploid in number and the megaspore mother cells will produce a embryo sac. The megaspore mother cells will produce 3 spores which we call as megaspores, and out of these 3 will get degenerate and 1 will get developed. After this, the microspores are developing and forming a pollen grains. These pollen grains are forming a pollen tube. This pollen tube will be having these 2 kinds of male gametes, and these male gametes, one is going to fuse with the egg cell and another is going to fuse with the polar nuclei, and after that the egg apparatus along with the male gametes will give rise to this zygote, and this zygote will form embryo, and this embryo is going to form a new plant body. So this is the life cycle of angiosperms. So the fertilizations which I was talking about in that life cycle, it is of double fertilizations. Now what is double fertilizations? Make it very sure that you understand this very clearly. What happening is, the male nuclei which is haploid is going to fuse with the egg cell. This egg cell is haploid. What is happening? One haploid male nuclei is fusing with one haploid egg cells. The diploid formation is taking place. This fusion is known as syngamy. After that, another male gametes, these male gametes will fuse with the polar nuclei. Well, polar nuclei are 2, that means they are having twice N, that means they are diploid. So what is happening? One male nuclei is fusing with 2 polar nuclei having diploid nature. This is forming triploid fusion, and the result is primary endosperm nucleus. The synergids and antipodals degenerate after fertilization, and then ovules develop into seed and ovaries get developed into fruit. This kind of fertilization is known as double fertilizations. So, I hope you have understood this part of angiosperm. What we have got in this part? The plants are having seeds that is covered and they are going to divide in two classes, dicot and monocot. Their embryo sac is 7-celled, 8 nuclei, and the pollen grains are having 2 male gametes. This 2 male gamete stage is known as 3-cell stage. You will get this in higher classes. So, these 2 gametes get fuses with the egg apparatus that we call as syngamy, and the next one fuses with the polar nuclei, which we call as triploid fusions. In this triploid fusion, they are going to form primary endosperm nucleus. So, this life cycle is very important. I hope you have understood. Now, let's see another part. A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through the voltage gated channels in the neuron's plasma membrane. Voltage gated channels opens and close and respond to an electrical voltage, so they are affected by changes in electrical charge around them. When neuron is at rest, a charge difference is maintained between the inside and outside of the cell, and this charge difference is produced and maintained largely by the active transport using sodium potassium pumps. The pumps and the sodium ions out of the cell and bring potassium ions in, while other channels allow some flow of potassium ions back out of the cell. The sodium ions cannot easily get back into replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The differences in charge between the interior and exterior of the cell is called the resting membrane, or you can also say it as the resting potential. When a nerve impulse begins with a stimulus, it disturbs the plasma membrane on dendrite and it causes sodium channels to open and the sodium ions flow into the cell, lessening the charge difference at that locations. If the change is enough, it will cause nearby voltage gated sodium ions to open. This allow so many sodium ions to flow into the cell at that location, so that the membrane there is depolarized with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage gated sodium channels, which then open, moving the depolarizations along the membrane, and this moving depolarization is called an action potential. These changes occur behind the action potential. To restore the resting membrane potential, the voltage gated sodium ions closes and the voltage gated potassium channels opens. This allows a rapid flow of potassium ions of the cell, repolarizing the membrane so that the inside is again negative and the outside is positive. So, you can see here, and this is followed by the use of sodium potassium pumps to fully restore the resting membrane potential and to re-establish proper concentration of sodium and potassium ions inside and outside of the cell. So, you can see here how the impulses are travelling from site A to site B and all throughout the length of the axon. So, this is how the action potential take place. Now, you have understood about this. Now, the next I will discuss about the transmission of impulses. That means how the impulses will travel from one neuron to another neuron. Well, how a nerve impulse is transmitted from one neuron to another neuron. It is transmitted through a gap, or also called as junction, called as the synapses. This junction is formed by the presynaptic node and the postsynaptic node. As you can see in this figure, the above part is known as presynaptic neuron and the lower portion is showing the postsynaptic neuron, and between this there is a gap. That gap is called as the synapses, and through this synapses they are going to transmit the impulses from one neuron to another neuron. There are mainly two types of synapses, that is called as electrical synapse and chemical synapse. In the electrical synapse, what happens that the membranes of the pre and postsynaptic neuron are in very close proximity provided by the gap junction between the two neurons, and in electrical synapse there is a minimal synaptic delay because of the direct flow of the electrical current from one neuron into the other across the synapses. And in such synapses, the impulses is very much similar to the impulse conductions along a single axon. These types of synapses are rarely found in our systems, and it is found in the cardiac muscles, smooth muscle fibers of the intestines and the epithelial cells of the glands. Whereas in the chemical synapse, the pre and postsynaptic neurons are separated by a fluid filled space called a synaptic cleft, and when an impulse, that is the action potential, is given to the presynaptic neuron, calcium ions from the synaptic cleft enter the cytoplasms of the presynaptic neuron, and this calcium ion causes the movement of synaptical vesicle to the surface of the knob, and these synaptic vesicles are fused with the presynaptic to discharge their content, that is the neurotransmitter, into the synaptic cleft. The neurotransmitter of the synaptic cleft bind with specific receptors, which are the protein molecules present on the postsynaptic neuron, as you can see in this figure, and this binding action changes the membrane potentials of the postsynaptic membrane, and it is done by opening the channels in the membrane and entering the sodium ions to the cell. This causes depolarizations and the generations of action potential in the postsynaptic neuron, and in this way the impulses is transferred to the next neuron, and the new potential which has been developed, it may be either excitatory or inhibitory. So I hope you have understood about this, how the transmission of the impulses is carried out. So it carries out from the presynaptic node to the postsynaptic node with the help of the synaptic cleft. That synaptic cleft will take the neurotransmitter, which is in the presence of the synaptic vesicles in the presynaptic node. So from there what happens, those neurotransmitter will get released into the cleft, that is to the synaptic clefts, and those neurotransmitter will taken by the neurotransmitter receptors, and then the impulses will move towards the next neuron. So I hope it is very clear with you. Now in the next topic I will discuss about the central neural system.